In this video, I'll be discussing the structure, functions, dynamics, regulation, and importance of HeLa cells. These cells aren't really widely known outside of the science community, however they gained more publicity when the book was published on them in 2010. I'm going to start by telling you the story of how they were discovered. They're named after Henrietta Lacks, who was an African-American mother living outside of Baltimore. During her pregnancy with her fifth child, she felt what she described as knots, and after childbirth she experienced abnormal bleeding. The doctor examined her and found that she had a lump on her cervix and sent this off to the lab. Upon analysis, it was discovered that she had cervical cancer. Despite several rounds of radiation treatment, the cancer spread, and in 1951 she died of the cancer, having only been diagnosed 9 months before. During her treatment, the doctor had taken tissue samples and had sent these off to a researcher called Dr. Gay. Henrietta had given consent for the treatment of her cancer, but she actually hadn't given any consent for the removal of the tissue. We'll be coming back to the issues related to this. When examining these samples, Dr. Gay found that there seemed to be cells that were replicating with no limit. These cells grew in vitro, which is in the laboratory, and these were the first cells to do so, as normally cells died within a short period of time. They were also found to have an unusual karyotype, which is the number of chromosomes, compared to normal cells. Normal cells, as indicated by the orange cells, experience the effects of aging over time. This is called cellular senescence. Repeated cell divisions makes the DNA unstable, possibly causing breaks or possibly causing toxins, and these are detected by the body and the cell undergoes programmed cell death. This occurs after approximately 50 cell divisions. Cancerous cells, however, don't experience this programmed cell death, as they have an active form of an enzyme called telomerase, which prevents the shortening of telomeres, the ends of the chromosome. The shortening of the telomeres is implicated in the aging process and thus cell death. It's also important to note that Henrietta's cells were especially hardy, even for cancer cells, and this wasn't really known why. It was suggested that it was possibly because she was also infected with human papillomavirus and syphilis. The uses of these cells were endless. Their stable growth led to the development of the polio vaccine. They're the first cloned human cells. They've been used primarily in cancer research, even to the extent that they've been sent to outer space to prove that cancer cells can grow there. They were fused with mouse embryos, which were the first cell hybrids, and these were extremely helpful in the mapping of the human genome. Also, their vast growth led to standardized protocols in the lab being developed. They've also been used in research in general, with 60,000 journal articles published of their use, and also in the field of virology. The field was essentially created by infecting HeLa cells with everything. So, are there any negatives associated with HeLa cells? The answer to this question is yes. They spread and grow quickly in vitro, just like they did in Henrietta's body, and because of this are called the lab weed, as they contaminate up to 20% of other cell cultures, which essentially ruins research. Also, there are serious ethical issues related to their discovery. This links back to the lack of informed consent. However, it's important to note that this was not unusual at the time, as it wasn't required. However, neither Henrietta nor her family knew that her cells were being used in this way, and initially when papers were published, the credit was going to an unknown identity of Helen Larson. Her family found out about the use of her cells in 1970 when her husband received a phone call from the hospital with someone telling him that his wife was still alive in the form of cells in a lab. The most important injustice seems to be that as the cells were commercialised, with a vial costing about $250, Henrietta's family were living in poverty with no healthcare and very poor education. Nevertheless, it's important to note that Dr. Gay made no profit from his breakthrough and actually gave the cells free of charge to other researchers in the name of science. More justice came to the family when an author called Rebecca Skloot befriended the family and published a book on Henrietta's story called The Immortal Life of Henrietta Lacks in 2010. Rebecca was also involved in helping Henrietta's daughter get into the lab where an iconic event occurred whereby her daughter held a vial of her mother's cells and whispered, "You're famous." Rebecca also created the Henrietta Lacks Foundation to raise awareness and also to help the family. HBO have now considered the book for a movie, and it's rumoured that Oprah may be involved.